education. Let me take you to an alien world beyond your imagination, where incredible things happen, invisible to you, but everywhere around you. I'm talking about photosynthesis, the process where water and carbon dioxide combine with sunlight to create oxygen and carbohydrate molecules. Life doesn't exist without it. We depend on it for the food we eat and the oxygen we breathe. Chlorophyll makes this happen, but chlorophyll doesn't just appear, as if by magic.
Not really. I'm going to take you on a journey into the nano world of chaotic collisions that allow a plant or algae or even a bacteria to build a molecule of chlorophyll from scratch, where what keeps our planet green and alive is powered by enzymes in a 16-step chemical process. But first, we've got to get to grips with how the planet-scale process of photosynthesis is all happening at the nanoscale in a whole different, very small dimension. We'll begin with a leaf, the basic unit of photosynthesis. We can see nature's solar panel. Give or take, this is about 10 centimeters. Let's start getting smaller. Part of it, 1 centimeter stem, is 10 million nanometers. The new unit of measurement, turn it on its side and go 10 times smaller to the thinness of a leaf at 0.1 centimeters, 1 millimeter, or a massive 1 million nanometers. Now let's go to the smallest thing we can see, the little hairs on the leaf. They're 100,000 nanometers.
Now we've reached our human visual limit, let's really get into this planet. Now we're at the level we can't see without a microscope.
At 10,000 nanometers is the size of a human cell, but we're not there, we're in a plant. We're in a palisade cell, squashed in with the chloroplasts. Think back, you might remember them from school. They're full of chlorophyll. We're now right in the powerhouse of photosynthesis, in a space about 30,000 to 40,000 nanometers. This is the structure our incredible proteins move around in.
Jumping down to their level. The nano world. We'll start with the viruses, who are getting all the attention nowadays. At 100 nanometers, they're the start of this exciting new dimension. But we've got to go smaller still. 10 times smaller, and we're with the proteins. We can go 10 times smaller again to a molecule, but we'll stick here with the proteins. They're the ones that matter to this story. They're the movers and motors that make photosynthesis happen.
This environment is incredibly frenetic, busy with proteins squishing past each other. Fortunately here, gravity doesn't matter anymore. We're not subject to it, there is very little friction, and things move at super fast speeds. Proteins can take any shape they feel like taking, whatever gets the job done. When the job is moving molecules in the process of photosynthesis, this shape can be like a wedge, a duck, or a trowel.
This world, with all its protein squishiness, doesn't just appear. It's all down to DNA.
DNA. Ugh. Right, so I know DNA is important. It's the fundamental molecule of life, and it defines who you are. Modern biology places a lot of importance on that genetic code. But for me, a human being is so much more than just a genetic code. It's a combination of thoughts, feelings, emotion, urges. And from a purely technical standpoint, fats, carbohydrates, and proteins swimming about in a load of water. You're effectively a walking, talking, sentient can of soup. DNA is apparently the recipe book for a human being, but that's not quite true. In fact, it's the instruction manual to produce proteins, and it's the proteins that go on to work or acquire all those other things like carbohydrates, fats, nucleotides, amino acids, micronutrients, vitamins, minerals, everything that you need to be a human being. Now that recipe book, that DNA, it does encode in terms of us a sentient can of soup, but with minor changes, you could then become a fern, a fish, a flatworm, a fungi, a ferret, and so on. This is what we rather grandly call the fundamental dogma of molecular biology, where you go from DNA to this stuff called RNA, which then becomes protein. Now, for our purposes, we can ignore this bit in the middle. and just talk about the whole DNA going to protein. Let me explain. A piece of that genetic code exits the nucleus, and it comes into contact with a rather large molecular machine. It's called a ribosome, and it's gigantic, biologically speaking. This machine latches on, and what it does is it reads that genetic code, passing along the DNA, and out the other end, spits out a bit of protein. And then, when it's done, it pops off. But at this point, you've just got a long line of floppy protein. And everything in biology is defined by its shape. It needs to fold. Now, there are three basic shapes in biology. There's random floppy loop, which doesn't actually have a shape, or there's a beautiful helix, and you probably will have seen that before with DNA, or there's a third one in proteins. And that is called a sheet. These are long protective regions that sort of protect the inner cores. So folding is an incredible process. We don't really understand a lot of it, and to be honest, if we did, then we would live in a utopia. We would have unlimited medicine, or the food that we'd need. But I don't know whether you've noticed, we don't live in a utopia at the moment. Now, even with our best supercomputers, we can get like a vague idea. Now, we have other ways of getting pictures, but what the protein is doing is it's folding up, hiding all of its water-hating bits on the inside and keeping all the water-loving bits on the outside. What it's doing is it's hunting for its most energetically favorable conformation. We call this falling into a thermodynamic well. And once we have a folded protein, then all the fun begins. Because this is biology, everything's a little bit squishy. And so, from one single fold or shape, the protein can actually change its conformation. Depending on whether it's really acidic outside or alkaline, whether it's really salty, or whether it's near a whole load of fat. And that's because there's a lot of conformational flexibility. Oh, look, it's a rope. So for loads of proteins, this is where their story ends. They do something structural, they hang about in the cell, they do their thing. But there's a special type of protein that I want to talk about because that's what I work on. Now, life, if you think about it, is just loads and loads of chemical reactions happening, thousands of them happening within your cells. Loads of these reactions, they don't happen fast enough for life to survive. They need to be sped up, and this is where something amazing comes along, an enzyme. Enzymes are the catalysts of life. They speed up chemical reactions. Let me show you how. Let's just take any old random enzyme. It's probably just sort of hanging out, floating around in the cell, gently pulsating. Maybe it's a bit more excitable. And then suddenly, maybe something that it wants to bind to comes along, and it changes its conformation. It binds to it, and this enzyme will then act on it and do something to it. But when it's done, it will reset, and then just float about in the cell again, doing its thing. I work on enzymes called molecular motors, and these are motors in the truest sense of the word. They take in a cellular fuel called adenosine triphosphate, or ATP for short, and they burn a chemical bond between phosphorus and oxygen. This produces something called chemomechanical motion, which is then transferred and then used to power a reaction. For example, muscle movement. But for this bit of the routine to work, it's pretty hard to wear a microphone when you're not wearing any clothes, so I've had to use a bit of movie magic and my psychic powers to tell you the story. Muscle movement is powered by molecular motors. And within your cells is an amazing architecture, a cytoskeleton made from a protein called actin. For your muscles to do their thing, a molecular motor called myosin grabs onto this skeleton, takes some of that chemical fuel, adenosine triphosphate, or ATP, and then burns a phosphorus-oxygen bond to produce a power stroke. And without molecular motors working consistently and precisely all the time, not only could I not move about in the air, I and you couldn't move at all. And what I find remarkable about all of this is that it's not just happening in me while I'm prattling about on this rope, but this is how all movement happens. And even when you're not moving, your enzymes are working, your molecular motors are doing things, your heart is constantly beating, your liver is removing toxins from your blood. This incredible dance occurring at the nanoscale within your cells is allowing you to survive and to thrive.
These motors aren't just one thing. It's a bit more complicated than that. They're a collection of proteins operating cooperatively together.
That's us. We build the planet's solar panels, pigment molecules. In nature, we are gorgeous, symmetrical, but random, and fabulous. A bit like this.
But in a lab setting, we have to be made synthetically. And for reasons yet unknown, things don't work out each time.
Oh, like that.
Or this time, and this time.
I'm CHLI, or I for short. I'm a motor which uses adenosine triphosphate to power a reaction. I take the magnesium ion and put it into the porphyrin, but I don't do this alone. CHLD over here,
You can call me D.
forms a bridge between us and our other friend CHLH, who couldn't be here today due to COVID-19 working rules and social distancing regulations.
H is my bit on the side, and loves to play with my feet. But this protein over here is my main squeeze.
Oh, hun.
Our interaction is much stronger. Want to play with my feet later?
Oh, do I have to? She always wants her feet played with.
Well, you can tell how much we like to hang out together because of our constant value. This measures the strength of our bonds with each other.
Our constant value is really high.
Play with my feet!
Oh, fine. We have a nanomolar interaction. In fact, it's so strong, it's almost a pico-level interaction. This means we're really hard to pull apart.
It's because I like having my feet played with so much.
So how much force exactly would it take to pull us apart?
I mean, I think we're pretty strong.
I think so too. Gun show. But before they could do that, they had to figure out how to peek into our nano world.
Perhaps.
I know, right? There's a few different ways they could do this. But we couldn't remember all the acronyms, so...
Science crew!
There is ITC, DSC, MST,
That's the one which I get to spray you with water.
No!
and of course there's the classic techniques like SPR or DSC, or a good old-fashioned native gel shift. It's not that hard.
The team from Sheffield decided to use a technique called a thump-fuck-load.
You want me to what now?
It's WAP now, and we'll do that later. No, atomic force microscopy, peak force quantitative nanomechanical mapping.
Oh, of course, it's so obvious.
Using some mirrors, a really sensitive camera, and some lasers, they can see the movement of the lasers reflecting off you as you vibrate.
Wait, they can see that?
Oh yes, baby. Vibing's nothing to be ashamed of. Everyone does it. As you move, the cameras can pick up the change in vibration. Oh, could you switch those off, please? Fine, the scientists managed to pull us apart. So what did they find out? How strong are we?
And drumroll please. The strength of interaction between I and D is 110 piconewtons, or 0.00000000011 newtons.
Is that good?
Well, two aerialists doing double trapeze exert forces around 4 times their body weight, which in this case is 2,500 newtons, which is about 23 trillion times stronger than 110 piconewtons. But for 2 teeny tiny proteins smushed inside the cell of a plant, literally powering all life on Earth, that's pretty good.
I thought as much. I mean, look at us. We're boss.
Gunshow.
So that chlorophyll molecule, how does it all kick off?
Chlorophyll is cool. It's the basis of all life. And without it, we wouldn't have this fantastic atmosphere. Without it, our planet would be a lot more, well, tumbleweed. If you zoom right out and look at the Earth from space, using your special binoculars, it's blue, yes, but it's also really, really green. As green as a pea on a plate. And all that green is chlorophyll. Now, Chlorophyll wasn't just kicking around waiting to become the basis of all life. Chlorophyll has to be made in a complex series of chemical reactions called the chlorophyll biosynthetic pathway. Every chemical reaction along the chlorophyll biosynthetic pathway has an enzyme to assist it. Now I think of this as a bit like how gravity helps us aerialists do what we do. Stay with me here. If I take the raw ingredients of this rope and my extremely muscular body, and I put them together in this combination, then add a little gravity enzyme, this is what happens. Enzymes assist the process by putting everything together in the right place at the right time. Now, let's zoom back in again to the very beginning of the chlorophyll biosynthetic pathway. Stop it. Or what we like to call the basis of all life. Stop with the drama. Life begins with a turns out it's not God after all. He, she, they do not exist. Life begins when a is inserted into a porphyrin molecule to make protoporphyrin 9. This innocent, squeaky-voiced little is the basis of all life.
But what about me?
You don't exist. Now piss off, you're ruining my show. Where was I? This is a really difficult and inefficient process. And nature only worked out how to do this once. It's a unique combination. Before chlorophyll, life happened in a reducing atmosphere. But with photosynthesis, oxygen suddenly filled the air, killing everything off dead in the first great extinction event. This magic reaction that created something different has only been worked out once. It really is magic. Now, once the magnesium ion is inside the porphyrin molecule, it has to be held very, very carefully to prevent it absorbing light and producing radical oxygen, which we all know from our beauty products is really bad. This would damage the cell, ruining the whole shebang. It also has to go on to make chlorophyll. There's no going back. And just like how without gravity that last aerial trick wouldn't have happened, without an enzyme, this chemical reaction wouldn't happen. The enzyme involved is magnesium chelatase. Stop it. Magnesium from the metal it works with, chelatase from the Greek to capture. The enzyme helps the porphyrin capture the magnesium. Now, not much was previously known about magnesium chelatase, although it had been studied a lot. Particularly, we didn't know where the porphyrin bound inside the enzyme or how the magnesium ion got there. So it was time for some structure function analysis. It's really juicy, that, isn't it? Really rolls around your tongue. The joy of structure function analysis. In biology, everything is based on shape. If you can see the structure, you can work out what it does. Just like an aerial. If you can see the wrap that I'm doing, and the position of my body, you can work out exactly what's about to happen. Images were gained of magnesium chelatase. We could see its structure, but we still couldn't see where the porphyrin bound inside it. A different approach was needed. It was time for a bit of virtual reality. Computational docking was used. Now, I don't really know what that is. Apparently, you give the computer your enzyme structure and its cargo, which is porphyrin, and you ask it where it sits inside of it. While the computer did that, I went off and had a good time on my rope. Whee! Whee! The computer came back with an answer, which then needed testing. We used genetic modifications to test our theories and rack up loads of experiments, introducing mutations to test the theory. So, what is the structure? Porphyrin is a ring-shaped molecule, and inside of it there are two dangly bits. These are sticky. They are there to glue the porphyrin into the enzyme. Once it's in place, there are two bits that point from the outside world all the way into the centre of the porphyrin ring. Like they are there for something to be put into, like they are waiting for something to arrive. Something like a magnesium ion, perhaps? One of them points from the bottom and one from the top. This is where we did all of our mutations. Now, when we mutated the bottom one, absolutely nothing happened. When we mutated the top one, it killed the protein instantly. Dead. That's how we knew it was quite important. We still needed more evidence, so we sent a crystallographer in to have a good look around. And she said she could see a channel that led directly from the centre of the porphyrin ring all the way to the outside world. A channel that the magnesium ion could enter through. It could slide in and take its place. So, bearing in mind that even in science there are no absolute truths, this is what we now think happens in the dance of porphyrin and magnesium. Or, how I like to think about it, how to seduce a magnesium ion. The enzyme is floating around in the cell. The porphyrin molecule comes along and it takes its time to slide into place. Once it's there, the magnesium ion approaches. Come on. Come on. In order for the magnesium ion to get into the cell, the enzyme burns a load of cellular energy. Basically, having a big extravagant party with some of its friends to allow the magnesium ion to slide into place. Once it's there, in the middle of this blaze of glory, it's transformed. and magnesium protoporphyrin 9 is released. The beginning of new life. The enzyme, never satiated, floats off to do it all again.